Aging is characterized by the time-dependent deterioration of multiple cellular processes that ultimately increases the probability of organismal death. Within a population, this process is variable and a number of genetic and environmental factors can affect it. One striking example is that of the disease progeria, a progressive genetic disorder that causes children to age rapidly. This disease is caused by a genetic change that alters the nuclear lamina, an integral component of the cytoskeletal network that underlies the nuclear envelope. This change leads to a loss of a post-translational modification, phenosylation, that ultimately leads to premature aging and a significantly reduced lifespan for the individuals who have this disorder. There have been many genetic screens that have identified genes that both shorten and extend lifespan. These include studies in the budding yeast, Saccharomyces cerevisiae, which is the focus of this methodology, as well as more complex multicellular organisms like C. elegans and the murine model system. These can be utilized to study several different aspects of aging, and we will be focusing on chronological lifespan, which is the duration that a cell can survive. The objective of this work is to provide a straightforward genetic approach to perform an overexpression suppressor screen.
Begin by identifying the genetic background that results in an abnormally shortened chronological lifespan, or CLS, in Saccharomyces cerevisiae using the Saccharomyces genome database. Select the Function tab from the options on the top of the web page, then select Phenotype, followed by Browse All Phenotypes. From the Yeast Phenotype Ontology options, scroll to the Development subheading and select Chronological Lifespan.